All right, so thank you so much for allowing me to talk for 2 hours here at this venue. My name is Danilo, Bzdok, medical doctor by training. I work both as a neuroscientist, as a quantitative analyst, as you heard, at Mila Institute, but also at McGill's Montreal Neurological Institute. So the mandate of my team is to really bridge the neuroscience activities at McGill and state-of-the-art quantitative analytics as practiced by Mila Institute. And so in the following, I'll start with a little bit of a conceptual primer and then give you just 3 examples of how we try to leverage large data sets using machine learning types of techniques to find new ways to answer classical questions. Quick disclosure. So what happened over the last 10 years since I finished university is that we saw an emergence of always larger, wider, deeper data sets. So the data set on the top here, 10 years ago didn't even exist. And that is a lot of structural, functional, white matter data from brains from a little more than 1,000 people. And we have maybe roughly 100 different variables, disease indicators, personality assessments, health indicators from these people. And at the time, that was when it came out, it was the largest data set at that point. So ever since the UK Biobank came in, and that is now the currently largest existing biomedical data set, at least in the open sphere. And if you haven't heard about it, this is 500,000 people from the United Kingdom that have a common variant genetic characterization, but also single base pair exome level information, as well as thousands and thousands and thousands of indicators from knee, tap, bone density to eye color. There's everything in this data set. And my team, this is our favorite data set at this point. However, and that's the lowest row, I think that in the near future, we'll see an emergence of always large data sets. It's probably going to be an exponential increase. And I think that some of the next largest data sets, they'll probably come from somewhere in Asia. So what does this mean for the types of conclusions that we like to draw about the brain and health using data? And here's a quote that I like very much, which is from Stanford's Brad Efron, and in 1993, he wrote that the biggest difference between the pre and post-war statistical practice is the degree of automation. So he refers to the fact that most of classical statistics, which is still the norm in biomedical research today, emerged in the time before electronic calculators and in a time when we had no choice than paper and pencil mathematics to come up with textbook formula as one-shot solutions to many of the quantitative analysis problems that we had. But ever since, and more than 20 years later, Brad Efron writes, almost all topics in 21st century statistics are now computer dependent. So, and what I will argue is that that's not just a technical difference, but also an epistemological difference in the sense that the questions that we can ask and the conclusions that we can reach, they are just drastically different if we are in the big data era. So here's an example that we published now 3 years ago with synthetic data of gene expression. Okay, so we generated data so that we know the exact data mechanisms, we know the solution, if you will, and we analyze these same synthetic data in 2 complementary ways. In panel A, you have the most common classical statistics, null hypothesis testing, p-value type of way to come up with which genes are relevant in my analysis, which could be in my disease research question. We then looked at the same synthetic data set with a more modern machine learning technique, which is random forest in this case. And the point is that the notion of importance that random forests are trying to get at is not the same. That's what I argue. And this is also what you see here in panel C. If you look for which genes have been identified as relevant, there's a divergence between the collection of most relevant genes according to a statistical significance notion of relevance and the predictive notion of relevance. So what this specifically means is the following. if in a major disease, such as, let's say, diabetes, we identify a certain gene to be statistically significantly associated with that particular population disease. That doesn't mean that the same gene is also the best we can do to predict outcomes in single patients. And that is, I think, causing a lot of friction and leading to a lot of confusion in the literature. We went even more in detail about this prediction inference dilemma. Here you see another empirical data simulation where we came up with more than 100,000 data sets that vary in the types of dimensions that we usually care about. Everyday data analysis, which is how much correlation is there in the data, how many variables are relevant, how much variation can overall explain the outcome. Maybe I already mentioned collinearity, maybe not. So they vary in all these dimensions that we care about. And we then analyze the same data set again. That's what you see on the left. Each dot is a pile of data sets. We analyze them in parallel with a linear regression approach to get at p-values, the best p-value for a particular data set. But then we also submitted the same data set to the most common way to assess importance in the machine learning world, which is out of sample prediction. So long story short, what we see here is something of a disconnection and a dichotomy between these two notions of relevance in biomedical data. Because if we find patterns in data that explain a lot of variation in new data points, then those data sets do not necessarily or consistently give us also at least one p-value that's statistically significant. But the opposite was true, however. If we find statistically significant results, then this does not always lead to predictively relevant information. Okay, so out of sample prediction accuracy appears to be somewhat of a more stringent criterion, at least in the way we have analyzed this here in this large scale data simulation analysis. Okay, so now 3 examples of what this means, what these types of changes in the questions we can ask and the types of results and their interpretations that we get. So here's example 1, and that's psychedelics. You probably heard in the popular press that there's an increasing interest in psychedelics now, especially over the very recent years. So why is it that we are currently interested in hallucinogenic substances? One way to see that is that since the 80s, in psychiatric treatment, there has been no essentially new drug mechanism or major invention. So we improved side effects of drugs against diseases like psychosis, schizophrenia, major depression, but there's no essentially different approach to any of the major psychiatric disorders today compared to 40 years ago. And recently, there were a series of randomized clinical trials in 30, 40, 50, 60 subjects that showed that if people take drugs like psilocybin, for example, we do see enduring benefit and relief in mental health symptoms, and that exceed those that are seen with classic drugs in this scenario. Okay, so that's really unexpected, because if this holds true, that could be the beginning of a major wave of new drugs in the space of psychiatry, which is a particularly important topic, in particular now, because as a result of the mass social isolation we're in right now, public health officials expect that there will be an increase in mental health problems over the next years. So how did we try to do something new now? And this is data from my own lab here. We did not run a randomized clinical trial in 30 to 60 subjects. We did something completely different. We looked at 7,000 text reports of people describing verbally in text, free-form text, how did they experience taking ecstasy, psilocybin, or other drugs. So our starting point for this analysis is literally only a series of text documents. But we can look at thousands of them, which is a big difference to the sample size that I mentioned in the clinical trials, which are logistically very challenging. Also, with this approach, we could look at 24 different drugs in the same analysis. And we also combine them with the specific neurotransmitter affinity mechanisms of each of these drugs. So the trick, if you will, of this study was really to realize that we know to what extent psilocybin and other psychedelics, they bind to dopamine receptors, let's say, in neuronal cells in the central nervous system. That is actually known, but it hasn't really been combined in this way. So we ran a pattern learning approach where we have 14,000 different vocabulary words on the one side, which is on the left here, and we have affinities of 42 different neurotransmitter systems and subsystems that we bring to the table at the same time. So, and we were surprised that this worked actually much better than we thought, because this first component of variation between experience, receptor affinity, covariation that we could consistently extract from this quite unstructured data, the first component looks like what people usually call ego dissolution. So ego dissolution in the space of psychedelic consumer experience is when people feel boundlessness. The boundaries between self and the world dissolve. Some people would say it has something to do with altering conscious awareness. And this is what we found as the most explanatory pattern across 7,000 reports and 24 different drugs, which is a scale that is going beyond the ways in which this question has been investigated before. So as you can imagine, this opens the door to really carefully quantify similarities and differences between all these experiences. So this is important because I haven't emphasized this enough. Because these clinical trials are so logistically challenging, we oftentimes cannot afford to investigate several of these drugs. So we can, for a fact, with the classical approach, not quantitatively measure very well how much they converge and diverge. What we also did, just to mention this is in the middle of the slide here, we then also involved yet another type of data to map it to the brain. So again, although we just start from text reports, we, in the last step of the analysis, even mapped it to regional differences in the brain of neurotransmitter receptor expression. So we know to what extent proteins for certain neurotransmitter subtypes are expressed in different parts of the brain, which is invasive data from humans. And the patterns that we find, they confirm what previously was reported in these clinical trials. And that is that we see somewhat of a gradient between higher association areas, the most recently evolved parts of the brain, and early sensory, more simple processing areas, if you will, such as the visual cortex. So this brings me to the conclusion for this first project that I wanted to mention here. And that is that combining text mining and physiological information of receptor affinity profiles, as well as regional differences in gene expression patterns, we come up with a fresh approach, a data fusion approach, to find a new perspective on why all these different hallucinogenic substances alter the balance between the higher integrative centers in the human brain and the early environment processing types of areas in the brain. Okay, second example, it's related but different. So schizophrenia is affecting 1% of the population across cultures. It's one of the most devastating disorders, and it costs millions and millions of dollars to our health systems. What we did here on this slide is a semi-quantitative text analysis where we look for which words do scientific papers use that talk about schizophrenia since the 50s. And we see that in the 50s, psychoanalysis was still an important topic in medical research on this major psychiatric disorder. This waned over the following years. We see that dopamine became always more popular in the 70s and remained an important topic in scientific papers on schizophrenia. But we also see that in yellow, for example, social cognition, social interaction emerged as an always more important topic in schizophrenia research. Why am I showing you this? That's to make the point that in my perspective, I think this is evidence that we are mostly following trends. So there are topics that become popular at a certain point in time, and then there's dozens and dozens of labs who go after these topics until there's a new hot topic that then dozens and hundreds of labs go after. Is there another way to maybe nominate, rank types of research, directions of research in a way that is maybe a little more impartial and data-driven? We try to make some tiny steps in this direction, and we did this in the following way. At the time when we ran the study, 500 subjects, sounds small maybe, was one of the largest schizophrenia data sets on the planet, with pooling data from multiple European, Northern American countries. We attacked that problem using a stacking approach. I can only summarize this here at a very high level. We, in a first step, generate a predictive model for a particular category of neuroscience experiments that people do in brain imaging scanners. For example, pain response, or speech tasks, or tasks that involve visual perception. So we have one predictive model that tracks information in the brain, specifically that where we know that their combination is tuned to a particular class of experiments that neuroscientists do. So once we have these predictive models, one for each type of experiment, we then train a meta model, a stacking model, which in this case is random forests, which build on top of these lower level predictive models that I have just described. And this higher level model can then rank which class of experiments is how successful in telling people with a diagnosis of schizophrenia and healthy controls apart. And this is what you see here on this slide. So on the top, you have those types of studies, which in general, we argue, are most likely to show predictively valuable results. We confirm several lines of research that have been ongoing in schizophrenia research for a number of years. For example, language processing, social interaction, which I mentioned before, we confirmed that. However, the top categories are types of experiments that haven't really been mentioned so much. And this is how we propose that there are directions of research that we may not have well thought out hypotheses for based on the existing literature, but just based on data. If we let the data speak for themselves, we find that they explain a lot of what tells schizophrenics and healthy people apart. And the same approach can be done in other major psychiatric disorders. It's by no means specific for schizophrenia itself. So just as a quick example, we can separate out these effects even more. That's the so-called partial dependence analysis. So although we have a highly nonlinear estimator on the top of this processing hierarchy that I talked about here, we can unbox the black box to a certain extent and look at the nonlinear surfaces and the discriminability of combinations of classes of experiments as well. And this is what I have exemplified here. All right. And the last topic I briefly wanted to present to you is about social isolation in the brain. So if you hear social isolation, or at least before the pandemic, it maybe seems a little bit like I'm going to say humanities type of topic, but we have known before that chronic social isolation and loneliness have pervasive influence not only on the mind but the entire human body. Individuals who feel lonely, they drink more, they smoke more, and they have various other types of unhealthy behaviors. Their bodies heal slower from wounds. Their immune system is compromised. Their sleep is disturbed, which in turn disturbs mental performance in their work life. I could go on. So this is a dramatic condition, even if it doesn't sound like that. And just to underline that fact, Japan, for example, has just recently decided to also have a minister of loneliness as part of the official government. And that's something that the United Kingdom has already done a couple of years ago, just to show you that this is really a quite serious condition and a big public health problem. So in response to the COVID pandemic, we have tried to make tiny steps in the direction of understanding what loneliness and social isolation really look like in the brain. So here we have structural brain data from 40,000 people. And what you see on the border is large-scale network level effects. So how do sets of brain regions that we know to belong to canonical brain networks that have been described before, how important are those explaining variation in healthy versus individuals who feel lonely? And you see that, and that's the top left, just summarizing this, there's several brain regions that really are part of the higher association circuits that are known to be subserving some of the most complicated things that the human brain is capable of, including social interaction. And that is something that we found here in this large-scale investigation, and that has not been described in the existing brain imaging literature on this topic before. Okay, here's just a model specification. We obtained the results in the previous slide by purpose designing a Bayesian hierarchical model that you can see here, where on the lower level, we have one effect for each of the brain regions that you saw, but we also have higher level priors with partial pooling effects, where we try to estimate the overall importance of sets of brain regions on the lower level in form of known canonical brain networks. And this is how we ended up with these variance components, these importance estimates in the panels B to H here. But it's a fully probabilistic extension of logistic regression, if you will. We also looked at the functional dynamics in the brain, what this looks like. This essentially confirms again that if we look at brain function, of functional coupling, intrinsic fluctuations of how brain regions spread across the brain are talking to each other, we again see that these highest integrative areas, this is really what explains a lot of the differences of how lonely people and not lonely people may be different. So just as one quick speculation, why is that? Why do we see these parts of the brain? What we argue in that paper is that we know in psychology that lonely people, they tend to talk more to their pets. It's really true. Lonely people are known to have overly intense relationships, imagined relationships with characters on TV. And lonely individuals are also known, have been known before, to remember more social events from the past or anticipate social events in the future, which may also be hypothetical. So why the highest integrative areas, which are so far away from the sensory cortices? Well, one idea is that lonely individuals may compensate for lack of social interaction in the real world by imagining, simulating, mimicking social relationships in their minds. And that's the most reasonable speculation that we could come up with to explain these data. So just as the last data slide, we then combine all these results from the various brain levels, brain structure, brain function, and we run what we call a demographic profiling analysis. So this is essentially a bootstrap difference analysis combined with a canonical correlation analysis. And it allows us to bring to the table 50 different indicators of health and behavior and predispositions that we know to be relevant in social isolation. And we let them compete against each other to find which of them explain best the multivariate pattern of loneliness-related brain changes. And we see here that, for example, several personality types play an important role in the experience of perceived social isolation, such as neuroticism. But we also confirm what I said before here in this brain behavior analysis, that sleep plays a role, lonely people drink more, smoke more, and so forth. And with this, just as a high-level summary, in health and analytics in particular, and maybe data science more broadly, here's some of the trends that we identified together with colleagues from Oxford University. We think that we'll see always more empirically flavored types of analyses at the expense of theoretically or purely theoretically motivated textbook formulas. That is what I talked about at the very beginning. Point two is that not only machine learning, but increasingly in health and neuroscience, we'll probably have large reference data sets that we use as test beds for analysis, but also for rigorous comparison of new methodological inventions. Point three is our classical notions of relevance and success, like p-values, they may change in importance. I'm not saying prediction is better. I'm saying there are certain questions, especially in precision medicine, with statements about single individuals, where there may be a lot of advantages for prediction, and where significance may be just not as well, not as good as of a fit. Point four is just that the analytical toolboxes have increased a lot over the last 10 years. And that has contributed tremendously to the democratization of statistical computation in academia and other sectors of society. Point five is just that new antagonistic paradigms in more academics thinking about data analysis may become dominant. So in the 20th century, we talked a lot about how is frequentist and Bayesian analysis different, and we may have new forms of antagonistic paradigms in 21st century data analysis in biomedicine and beyond. And with this, I thank my collaborators for the inspiration here on the left. I thank the institutions on the right for the financial support, and I thank you for your attention.